The overall goal of this procedure is to quantify the superoxide, hydrogen peroxide, and NADH production rate of purified flavin-containing mitochondrial dehydrogenases using microplate fluorometry. Mitochondria can contain up to 12 different superoxide hydrogen peroxide forming sites, which have been identified using different substrate and inhibitor combinations with isolated mitochondria. However, some difficulties still remain in regard to accurately quantifying the release capacity of individual sites of production in isolated mitochondria. And this is associated with unwanted side reactions that also produce reactive oxygen species, but also the presence of antioxidants and the use of inhibitors that lack specificity, in addition to the use of additives that can interfere with measurements. Here we present a method for the simultaneous measure of superoxide, hydrogen peroxide, and NADH production by purified flavin-dependent mitochondrial dehydrogenases. This method is advantageous since it can allow for the direct and accurate quantification of the ROS-producing potential of individual sites of production by eliminating unwanted side reactions and any reactive oxygen species quenching systems. The simultaneous measure of both ROS and NADH production also allows for a direct comparison of the superoxide hydrogen peroxide forming potential and activity of individual enzymes, which can be valuable for screening inhibitors for ROS production prior to conducting assays with mitochondria. For our purposes here, we will be using pyruvate dehydrogenase complex and alpha